Alright, so in this video I'm going to be talking about another approach for biomedical event extraction. Again, we are using generative language models. So I'm going to be talking about this paper in 2023, which uses this idea in 2020, text-to-text transformer. So the recent success of sequence-to-sequence language models like T5 brought noticeable improvements for many general domain natural language processing tasks. However, so far these models found little application for complex domain-specific problems such as biomedical event extraction. Biomedical event extraction is a challenging task with the goal of extracting complex structures that describe the relationship between multiple molecular entities from scientific text. The structure of biomedical events is similar to a graph, which makes it non-trivial to decode structured prediction from a single sequence. So Paolini and his colleagues presented a framework called transition between augmented natural languages to solve such structured prediction tasks using sequence-to-sequence language models. It generates sentences for solving tasks like relation extraction, NER, and event extraction. I have a playlist for RE, NER, and this current playlist for event extraction. So in this paper, we investigate the effectiveness of this framework for biomedical event extraction. We design a natural language description to solve biomedical event extraction in sequence using this framework, and evaluate it based on T5 language model. Like previous lecture, again we use T5 language model. Our results show that a generative model can perform the biomedical event extraction. However, our approach does not outperform the baseline on any test data set. We find that our model struggles, especially with the argument detection step. So this is a sentence, you see, it contains two events. The first event is a gene expression event, in the yellow here, with the protein entity blue as its theme argument. But the second event is a positive regulation. Here is the second event, and with the protein entity LMP1 as the cause of that cause argument. And the gene expression is just the theme. So the theme of it is by itself an event. So it's an example of nested event. The processing is like this. First, we get the data and then we give it to this language model. So we have some input and we have some output. But in order to facilitate the sequence-to-sequence model, the inputs and targets of a given task have to be encoded into sequences in TANL. This natural language description is based on a markup language where task-specific information is highlighted with square brackets. It follows a strict pattern to later be able to extract the information easily. The framework provides multiple adaptations to solve a variety of information extraction tasks. So as I said, Paolini proposed an adaptation for event extraction that covers data sets with a small number of event types and flat target structures. However, this adaptation does not support nested event extraction and higher order event extraction. Nested events are events that have the same event trigger but different event types or arguments. Events with other events as an argument are called higher order events. Both types are more difficult to extract because the model also needs to consider knowledge about other events in a sequence. However, typical biomedical event extraction datasets contain deep event structures, which means nested or higher order events are very common. We therefore extended the NLD for BE task to support nested event extraction as well as higher order event extraction. So we modified the input and output to generate task-specific input and target sequences. We tagged the given goal entities in the sentence with square brackets and added the corresponding entity type as context or model inside the brackets, as this is the standard for BioNLP evaluation. The entity name and the type are separated by pipe character. The target sentences are annotated by framing the event trigger in square brackets. Inside the brackets, the event type and the arguments are added and separated by pipe. We use a nested pattern for the event extraction target sentences similar to the nested entities proposed by Paolini. With this modification, multiple events can be extracted from a single sequence. So the function can handle simple and nested events, but around 10% of the generated output have different text than the original sequence. Such reconstruction errors are documented over various datasets. For example, here the model generated increasing instead of increase as an event trigger. So especially incorrectly generated triggers can cause entire events to be incorrectly predicted, reducing the accuracy of the model. To repair such cases, string alignment is implemented based on this algorithm, Wunsch algorithm. And the dataset for obtaining train views Big Bio framework, because it enables easy programmatic access to over 120 biomedical datasets. With that framework, we could access all used BE datasets in a unified format. So this is very important. If you want to do event extraction on biomedical text, why not using this framework? And so this is Genia dataset, and